Good evening, examiner. My name is Dang Ziqi with matrix number of 103763. My title is Evaluation of CRISPR-Cas12a mediated stop codon insertion in the PSL gene to reduce biofilm formation in Pseudomonas aeruginosa. The microorganism that I choose here is Pseudomonas aeruginosa because it is a major biofilm producing bacteria and recognized by WHO in 2017 as the most lethal bacteria. Therefore, WHO listed this bacteria as the primary research target for antibiotic development. The virulence of this bacteria comes from the ability to form biofilm on biotic and abiotic surfaces that will lead to antimicrobial resistance in disease (AMR). Scientists have estimated that if no effective approach taken towards this AMR disease, it is estimated to cause about 10 million of deaths by the year of 2050. According to previous research, PSL is recognized as the main protein that contributes to the biofilm matrix formation because it can stimulate the initial attachment of bacteria to the surface. The objective of this research are to design cRNA that will targeting the PSL gene of Pseudomonas aeruginosa, to introduce a premature stop codon into the PSL gene using the CRISPR-Cas12a system, and to evaluate the disruption of PSL gene on biofilm formation in Pseudomonas aeruginosa. The first step of this research is to engineer a precise CRISPR-Cas12a system. By doing so, first we need to identify the PAM site of the gene with sequence of 5' TTTV3'. Next, the cRNA is designed approximately 20 to 24 nucleotide which complementary to the non-coding strand. On the other hand, a donor DNA with 50 to 100 nucleotide is synthesized with stop codon TAA inside. The stop codon TAA is positioned between left and right homology arms with 25 to 50 base pairs each. The final step is to prepare three different plasmids, which include cRNA expression vector with cRNA and gentamicin antibiotic resistant gene inside, PBBR1 MCS derived vector with Cas12a protein, sigma red recombinase, and kanamycin antibiotic resistant gene inside, and non-replicating plasmid with donor DNA template inside. The second step of this research is bacterial transformation. The three plasmids are first transformed into Escherichia coli because of its non-biofilm forming ability, which is easier to transfer plasmid than Pseudomonas aeruginosa. After that, the transformant is selected in LB agar containing kanamycin and gentamicin in order to select the successful transformants. The successful transformants is then incubated with Pseudomonas aeruginosa in another LB agar to allow conjugation. The conjugation is the most important part as the E. coli will transfer the plasmid to Pseudomonas aeruginosa through reproductive pili. The bacterial sample is then collected at log phase to further culture inside a PIA agar, also known as Pseudomonas isolation agar, with the purpose of eliminating the remaining E. coli in the sample. The third step will be the induction of CRISPR genome editing. From the PIA agar, the Pseudomonas aeruginosa is cultured. Then, inducers are inserted into the solution to initiate the CRISPR action, as the CRISPR will target and cleave the PSL gene and stop codon will insert into the cleavage. After the induction occurs, the bacterial culture is transferred in LB agar to let the bacteria heal and grow. After that, the colony's PSL region is then amplified by PCR and Sanger sequencing is used to verify the insertion of stop codon TAA in the PSL gene. After confirming the presence of TAA inside the genome, the bacterial colony is then cultured inside antibiotic-free media to allow the natural loss of plasmid, and this is the plasmid curing process. The last step will be the biofilm formation assay. Two different assays were used in this research. For the crystal violet assay, the wild type and the mutant strand is grown to let the biofilm form. After that, PBS is used to remove the bacteria and left the biofilm only. Then the biofilm is stained by crystal violet dye and measured in microplate reader to see the quantity of the biofilm formation. Next, CRSM assay is used by growing both wild type and mutant strand on a glass covering slip to allow biofilm formation. After that, the stains dyes the live bacteria, the dead bacteria, and the biofilm exopolysaccharide. After the CLSM microscope obtains the image is sent to COMSTAT to quantify the characteristic of biofilm and bacterial form in the image. Figure 2 shows the result of the biofilm formation of the wild-type incomplete gene expression mutant and the complete CRISPR-edited PSL mutant. The first image shows continuous green layer covering almost the entire surface, means the wild type do actually develop dense and mature biofilm. The second image shows that the biofilm is still present but appear less, which indicates biofilm forming ability is still there, but the biofilm produced are weaker than the wild type. The third image shows very little green color, which indicates the PSL disrupted mutant do exhibit poor surface attachment and limited biofilm formation, and they show positive result as preventing the development of a mature biofilm. Therefore, for this research, all the objectives are achieved.